the wave particle theory of brain dynamics. And no quantum mechanics will be used. This is a theory of large-scale systems. I did begin by thanking the organisers, so I'll get going. There's four parts to this talk. It's quite a large talk, and there is some mathematics, but I've endeavoured to explain everything pictorially as well. So some basics of dynamics, then a segue into whole brain neural mass models, neural field models, which is this wave-based approach to modeling brain behavior, and then a reconciliation of these two different approaches, one being a wave approach, one being more a particular particles and networks approach. So some preliminaries. So, I mean, at the heart of what I do, and what are those of us doing biophysical and computational forward modeling, is an evolution equation. So this is an underlying model of neural states. This distinguishes us from, say, machine learning, which are sophisticated statistical models of the data. These are models of underlying states which generate or cause the data. So I'll just talk through this very generic form of an equation. On the right-hand side, we have this function f. That puts all the physics of the system into this equation. And then we have I, which are the inputs flowing into the system. X is a state vector. This is a vector of all the states of the system: firing rates, membrane potentials, ion currents, all the beautiful underlying neurophysiological states. The parameter A is a control parameter, something that it could be manipulated externally. It could be temperature, it could be the bath concentration of an ion. And then on the left-hand side is the famous dx to t, the integral, the differential, the derivative. And this makes all of these states change in time. So this is an algebraic appearance, but these have in parallel geometric forms. So in this representation, X is a phase space. So it's a Euclidean manifold, and I and F then defines a small vector at every point of this manifold X. So we get this whole flow of vectors. Then we can piece these together with solution curves or parameters, and these flow onto things called attractors. This is physics and mathematics, which is hundreds of years old. So here we've gone from an equation through to an attractor. The attractors come in different forms. They can be closed, they can be loopy, and on the right hand, we have something called a chaotic or strange attractor. And there's a mapping from the topology of these attractors through the type of time series that one might observe if one put an electrode into the system. So simple periodic, then something that looks like bursting with a slow current, and then mixing of timescales that give rise to chaos. Just as an example, this is a model of a small population of excitatory and inhibitory cells by Hugh Wilson and Jack Cohen. Now it's quite famous. These are the two equations. The states are capital E and capital I. On the left, we've got the phase space with some vectors and a detractor. And what we're going to do, and then we've got the time series, is we're going to, as our parameter, manipulate up and down an external current to the excitatory cells. So that's this JE term. And what's really important here, there's a few features, is the phase space, the vectors change smoothly and very slowly with the parameter. But the underlying or the resulting orbits can change very dramatically. Here we see it going from a fixed point to a limit cycle. This is called a bifurcation. So this is a step forward from flows to attractors. This is what a bifurcation looks like. I've put the parameter now on the x-axis of the bottom figure there, and I've got some cut-through cross-sections on the left below the critical value. We've got a fixed point, then we've got a very small limit cycle, going up to a large and ultimately deformed limit cycle. This is a canonical bifurcation called a Hopf bifurcation. And many neurons in the brain fire through a Hopf bifurcation. And that's not the only type of bifurcation that makes neurons fire, but it's one of the core dynamic mechanisms to get a neuron firing. So here we go, just demonstrating the Wilson-Cohen model. I'm sweeping this external parameter up and down, and you can see the onset and offset of these oscillations. Oscillations obviously are ubiquitous in the EEG, and this is like, if you like, the dynamical origin of oscillations. So Hodgkin and Huxley, through careful observation and manipulation of the squid giant axon, wrote down a set of dynamical equations and won the Nobel Prize for this in the 1950s, the famous Hodgkin-Huxley model of a single neuron. And it's based upon, I don't want to go through the equations here, there's no time, but a capacitance model of the neuron with potassium and sodium channels being voltage gated. And they showed through the interplay of fast sodium and slow rectifying potassium, you could explain why a neuron exhibited an action potential. And they had very detailed models. And we know even simplifications of this model can reproduce the whole repertoire of how neurons fire. So I talked about Hopf bifurcations. There's other bifurcations, saddle node bifurcations, etc., that this model derived from neurophysiology can exhibit. But individual neurons are buried within circuits of thousands of neurons, which are themselves immersed in functional subsystems and large-scale networks. Individual spikes are very weak, and they're in general, not always, but in general, they're too weak and too noisy to influence the sort of global macroscopic perception and behavior of us as agents. We have billions of neurons, and the body, for example, has a relatively low dimension of subspaces that it moves in. So it seems more likely that it's populations of neurons firing together that underlie the coding principles of brain function. And you can think of each neuron contributing like a vote to that population ensemble. We'll show that in a little bit more detail here. Here's a lovely microscope vision of some neurons. We can code those up into simplified spiking neuron models that I've shown here. And if we've got 1,000 of them, that's a 2,000 dimensional system. But we're not going to try and understand that. If we take two cross sections through these spike rasters showing all the spikes, we see two different times, two distributions that look approximately Gaussian. So we can do something called a mean field approximation, and we can get rid of all of those high dimensions, and we're just left with the mean and the variance. So that's the essence of a mean field approximation. Then we can use a different type of equation that just propagates this distribution forward in time. And if the distribution is propagated to match the actual distribution, then we've got something that seems to be working. This is the heart of generative models of neural populations. And this is widely used in physics, but is it even relevant at all in the brain? And we've just published a paper that I'll walk very quickly through now, or about to publish, that I think provides some evidence for this bridge between neurons and systems. And many of you may have seen these beautiful videos, should be whole field, not wild field, calcium imaging in lava zebrafish. This was published in Nature Methods a few years ago, 2013, sorry, and these are single unit recordings. So what Max Shine and Brendan Munn did, and you'll find this on Archive or bioRxiv, is to take pairs of spikes, and spikes are very sparse, so they're definitely not Gaussian, and greedily pair them, and then take those duplets and pair them again, and so on and so forth, right up until where every neuron has been paired, and we've now got macroscopic compilations of individual neurons. And what you see on the right-hand side is very important here, that the distribution goes from being very sparse or very kurtotic to being broader and broader until just below the macroscopic state where it becomes a Gaussian field. And Brendan and Eli and Mac and I reproduce this in all the different large-scale recordings that we could get in the open domain, and that hopefully will be published very shortly. So that's the underlying logic of going from neurons to populations and the essence of how to do it mathematically. So this is what I've just walked through. We can do this at any scale. In fact, I mean, a single neuron model is a mean field approximation of underlying currents. And then we can put them into compartments, we can lump them into neurons, and then we can lump those into whole populations, such as the Janssen-Ritt and Wilson-Cohen models. Now, we don't lose all the underlying neurophysiology. Basically, if the distribution of the states approximate these Gaussian distributions, then through some tricky mathematics, when we change the underlying neurophysiology, for example, through neuromodulation, through catecholamines, through noradrenaline, then we can map those single neuron manipulations up to the large-scale mean field approximations. So we're not losing the neurophysiology, we're averaging over it, but we can still have the parameters from the neurophysiology mapped and charted up into the population level. So now essentially a toy model of a neural population. This one's got a pyramidal cells, inhibitory cells, and potassium channel conductances. I'm not going to write down the equations, but there's two fast parameters and one slow parameter. And when you change the, depending on your the rest of the mixing of these two timescales, you can get either a bursting attractor, this is a quasi-periodic attractor, or you can get this interesting looking a chaotic attractor. It's actually called a homoclinic because there's one fixed point around which the dynamics are organized. So this would be a model of a single population in one part of the cortex. Now we're going to start taking other populations and pairing them together. So we're going to allow populations to influence each other. And that's this function on the left, G, which combines these two populations. And how do we do this? We've been able to take, in the first years, we took compilations of direct tracing from COCOMAC, and more recently, we use whole brain tractography to reconstruct these connectomes. So these are the underlying structural connections. So we have a little attractor. In this model, I'll show 500 times. We couple them together, and then we can perform systematic numerical simulations and see what happens. So a whole lot of people of us worked on this. I know Pedro has been very active in this. Of course, Vic Yersa, who I saw on the program, Usteko, Olaf Sporns, and others. This is a paper that we published a few years ago where we observed at the very largest scale all these wonderful exotic looking traveling waves. The point to make here is that I mathematically forced a mean field reduction from thousands of dimensions to 2 or 3 dimensions in each node. Then the underlying mathematics is finding new dimension reductions, natural forms within the flows, taking thousands of dimensions and finding relatively low dimensional underlying manifolds. When I say low dimensional, I mean maybe 12 dimensions. I'll show a little bit of this in a moment. Waves some years ago could have thought of being to be exotic, but they've been observed in different recording modalities across different species for different functions in different areas: motor cortex, visual cortex, epilepsy, sleep, whole brain, you name it. There's a whole explosion of wave-based observations. Why do these neural mass models exhibit these waves? It's because and I'm going to come back to that, at the heart of the connectome, it looks like a very complex network, but individual pairs of nodes that are nearby, that's the distance on the x-axis here, tend to have very strong connected weights. And as you get further and further, the strength of those weights gets exponentially weaker and weaker. And this underlying synaptic kernel pushes wavefronts along. Here's some recordings that Terry Sanowski's group published in eLife during sleep in people with epilepsy. And here's this relatively toy model that I've just shown. The same sort of timescales, the same sort of wavelengths, and the same patterns of waves. This is definitely not model inversion from data, though. This is a proof of principle. Where do these different waves come from? To understand this, we did a little bit more dynamics, and we noticed that one wave pattern will transition to another wave pattern and then on to another wave pattern spontaneously. So you can partition out the flow, which is the x-axis here, into these discrete metastates. To get a little bit more insight, each node we can look at a vector of how close its neighboring states are. That are these vectors. So we're sort of going back towards the vector field, and then we can use streamline technology to find the underlying orbits. And so this is what you see here. On the left, a wave pattern, in the middle, the vectors, and on the right, the underlying streamlines. What you see, these fixed points, nodes, sources, and then these vortices. So it's really a type of spatiotemporal chaos. And when the sources and sinks collide, you get a transition to the next state. Just a few years ago, even in fMRI bold data, people have started to observe these whole brain wave patterns. Right, so at the essence, however, the neural mass model is composed of individual nodes or particles connected together through the connectome. And that is an approximation that has worked very, very well in the last 15, 20 years. But even though the brain has this, the connectome has this beautiful network that Olaf and others have characterized, this is what I was talking before, that if you plot the length between two nodes on the x-axis and log of the number of streamlines corrected on the y-axis, you get a straight line. So that's an exponential underlying exponential distance rule, which I've written up here. Remember, I talked about this G function, which you can see in the equations, and it can be approximated as e to some exponent sigma times the distance between the nodes for some constant sigma. Now, about 10 years ago, James Roberts and I developed an algorithm where we said, well, this is a beautiful connectome, but it's spatially embedded, and how much of this connectome just comes from the spatial embedding? So we developed a technique to create connectomes that only had the exponential distance rule. And then we put them back into the brain, and you observe quite a lot of the clustering, quite a lot of the small worldness. It's not the full, beautiful connectome that Olaf and others have demonstrated, but you get about 80–90% of the way there, just through the exponential distance rule being spatially embedded back into the cortex. This exponential distance rule isn't an artifact of diffusion-based connectomes. Henry Kennedy's group have found it in direct compilations of mouse and macaque, and this is our human inferred connectome on the bottom. So it's a common observation across species. So this lies at the heart. So to do a neural field model, we're going to really dig into that exponential distance rule. And this is how we're going to do it. We're going to say two elements in a smooth cortex. We're not having nodes anymore. This is a smooth field, are connected both in the black line, which is within the cortex, and in the red line, which is through the white matter, by this synaptic kernel. Now I've called it capital W. Vic Yersa, Herman Harkin, who recently passed, Peter Robinson, in the 90s showed that you could put this into a very, very general integral equation where you've got the field phi on the left being an integral of the firing rate at different sites across the cortex, arriving through this connectivity kernel. We're not imposing anything else on the model at the moment. An integral equation, as the mathematicians will know, is a much more general form than a differential equation. So now we're going to put into the heart of this synaptic kernel this exponential distance rule. We're getting rid of all the noisiness. We're just taking a strict exponential distance rule. And we're putting that into this W, into the equation. Now, when we take something called the Green's function, you're going to have done applied maths or physics to know the Green's function. It's some fancy maths with the Fourier transform. We can go from an integral equation back to a differential equation. So this is a single differential equation now for the whole cortex. We don't have 500 differential equations at each node. We have one differential equation for the whole cortex. So this really is due to Herman Harkin, Vic Yersa, Peter Robinson. And there are others, of course, and Pedro would correct me because he knows the history much better than I. I'm going back, but these are the sort of wave equation models. And this upside down Laplacian squared is the spatial derivative, the second spatial derivative. It's the curvature of those vectors. It's not the vectors themselves, it's the change in the vectors. It's a second spatial derivative. And then with Peter Robinson, we're going to put the thalamus in. So there'll be a relay station from the cortex to the thalamus, and that goes into the differential equations. So this is now a wave equation for the brain. And last year, James Pang, Alex Finito, Kevin Aquino, Peter Robinson, and I did this whole survey of just what you can get out of this wave equation. And it's almost 100 years since the Schrödinger wave equation of the atom. It's actually similar but different. There's a second order time derivative in ours, but you can ping the cortex and you can see the propagation of the perturbation across the cortex. Now, this is a lovely animation by James. So what do these neural fields do? So I'm going to take out the spatial derivative. We're just going to look at the temporal derivative now. It's a damped harmonic oscillator with a time delay. And we can do quite a lot of mathematics with that. We can also put that into MATLAB or Python. It's a form of a damped harmonic oscillator with a time delay. And what you can do with it is quite interesting. You can now put it into bifurcation theory. Here's a 3 Hertz bifurcation. And this predicted or reproduced or explains very beautifully the 3 Hertz absence seizures that you see in many juveniles. So both the onset, the waveform, the power spectrum, the symmetrical appearance of petit mal seizures or absence seizures. And then you can go and look at a 10 Hertz. It's a slightly more exotic Hopf bifurcation. It's called a subcritical bifurcation, and it's now got a zone of instability. And this looks very much like a grand mal or tonic-clonic seizure, both in terms of the onset and the offset, and once again, the seizure. What you don't get is the slowing down of the time scales as naturally as you see in nearly every generalized seizure. And that's work that we've been focusing on more recently. So it works pretty well. What about spatial processes now? We're going to put this Laplacian back in. And to get some intuition about how to do this, we're going to go back to physics and back to a very famous system called the Chladni plates. This is a plate with sand on it. You just vibrate it. And when you vibrate it with different oscillations, you see the sand going into very beautiful harmonic patterns. Depending on the frequency that you put in, you see different patterns. Now, what the sand is doing, if you think of a standing wave, the sand is going away from the peaks of the standing waves and along the nodal lines of the standing waves. So it's really a way of visualizing the underlying harmonic patterns in this vibrated rectangular plate. Now we can write down a wave equation for a rectangular plate, depending on its stiffness. It's a form of Newton's law, F equals ma, with everything else that goes into being a metallic plate. And we can do Fourier series, Fourier decompositions, two-dimensional ones, and we get these eigenmodes. These are the natural waves, manners in which this plate will vibrate. And I've just shown some low-order ones, but there's higher and higher order. Just like you get sine waves in the EEG, you get these sinusoidal harmonics in this geometric shape. So that's exactly what we're going to do with the brain. And it sounds crazy. We're going to write the wave equation in the Fourier domain. Don't worry about the details. And then we're going to break it into the spatial modes and then their temporal behavior. This is a very common way of dealing with these types of systems in physics. And so we've got these fields being composed of these spatial eigenmodes Y, which vary over the cortex R, and then a dynamic, which is e to the minus omega t. So they have a natural frequency, and I'll talk to that in a moment. The eigenmodes can be derived by solving this Helmholtz equation, which is getting rid of the time, on the cortex. It's a form, of a singular value decomposition, and they're like spherical harmonics, but they're adapted to the shape of the cortex. And there's now a number of Python and MATLAB algorithms for doing any shape. And these are the harmonic modes of the brain. So now as you go into higher spatial frequencies, they would be paired with faster temporal frequencies. So the global mode in the top left has the slowest frequency, and the higher modes have faster frequencies. I'm not going to talk through why, but that's the way the decomposition works. So solutions to the damped wave equation are superpositions of the excited eigenmodes plus some noise. So when we want to look at a solution, we've got the eigenmodes and they're scaled up with a scaling coefficient. So just to write that geometrically, here's a pattern on the cortex. It's going to be a sum of the eigenmodes, each with their coefficient. And then there'll be a carrier frequency for the different modes. We used this a few years ago to explain neonatal EEG, but I'm going to walk backwards and show some fMRI and some other EEG examples in the last few slides on this. But these are the spherical harmonics that were solutions to the Schrödinger wave equation for the hydrogen atom, and these are the geometric harmonics, which are solutions to the same sort of principled approach to the cortex. So now to show how it works in real time, I'm going to show a solution of this by one of my physicists, Richard Fogat, who's joined me last year after finishing her PhD at the famous Indian Institute of Technology. And this is a full-blown numerical integration of these eigenmodes now. And because of this thalamic loop, when you look at the power spectrum, you get this large alpha oscillation. And to my knowledge, this is the first corticothalamic neural field simulation, at least shown in its full richness like this. Richard and I are working on seizures, so hopefully you'll see more of this soon. But what about task fMRI? People might say, guys, you're crazy. This is lovely theory, but this doesn't really mean anything for the fMRI signal. So classically, whole brain fMRI is broken down into individual voxels, which are then subjected to mass univariate general linear theory. And then we get these thresholded effects, as we know, or blobs. But we're going to instead, and that makes the task effects look very isolated. We're going to look at this eigenmode analysis. We're going to reconstruct these whole brain maps from eigenmodes. We went to the Human Connectome project, I should say James Pang, Alex Finito led this. We took all these different tasks and we reconstructed it as linear combinations of different eigenmodes. And this is probably the most important slide from that paper. Going from left to right, you see you very quickly reconstruct the fMRI maps. By 10 modes, you've got 50% of the data. By 100 modes, you've got 80 to 90% reconstruction accuracy. Now, this is important because the 100th mode has a wavelength of 6 centimeters. That's saying 80 to 90% of the energy in fMRI is at scales of 6, 7, 8, 9, 10, up to 20 centimeters. The small scale effects we see are right out. I mean, this is a 30,000 mesh vertex reconstruction. So there are 30,000 modes, but we only need about 200 of them. And they're all the large scale modes. We benchmarked against other other ways. You've probably heard of connectome modes, which you can get from doing a Laplace decomposition of the connectome. From the Human Connectome project, the connectome modes did a much poorer job than the geometric modes reconstructing. They still do an excellent job. If you add a lot of additional pre-processing, then the connectome modes will start to approximate the geometric modes. But now we're talking about things like smoothing of the connectome within the cortex itself. Don't forget these geometric modes, all you need is a T1 weighted image. So this is great for you, Pedro. You don't need, this is cheap, relatively cheap. It's a 7-minute structural scan to get these modes and then infer from EEG data. So we also benchmarked the neural field model against these old neural mass models. I'm not going to walk through this very complicated slide, just to reassure you that this simple neural field model with one free parameter matched or outperformed the neural mass model with its thousands of parameters. I mean, you can fix all the parameters in all the nodes to be identical, and then you've got about 8 free parameters. So I'd worked on neural mass models for 20 years, so I was quite happy to see it beaten by a neural field model. Not everybody was happy, but the nature of science is that different fields have their own approaches and rise and fall. And these propagating waves from visual cortex, they get bigger and they slow down, and that very nicely recapitulates what's seen in receptive fields in primate visual hierarchies, that they get larger. This is one of the canonical thing about visual receptive fields in the brain. As you go up the visual streams, they get larger and slower. And this neural field model will do that. Okay, now what about a reconciliation of fields and masses? Now, I said you could get a long way by averaging a lot of the detail out of the neural mass model, out of this particle approach to the brain, just with a simple exponential distance rule. So the first order, yes, all you need is this exponential distance rule in your neural field model. But what is missing is the heterogeneity. The real connectome is not a pure exponential distance rule. It's a noisy exponential distance rule. And most importantly, some connections are much, much stronger, 2 orders of magnitude stronger than you expect them to be if it was just an exponential distance rule. And here we've just chosen and highlighted 2 very long connections in the cortex that are much stronger. So these are anisotropic. These are atypical connections. So what do we think that we're doing? Well, Peter and I have been going to the brain connectivity workshop for many years. And Vic Yersa presented this when we came to Cuba. We came to Havana and had a wonderful scientific summit in Havana in 2002, I think it was. And Victor presented some of this work here. I won't talk through the details, but this is the homogeneous. You see homogeneous, this W-HOM. This is the exponential distance rule that is in this neural field model. And this yields these stable damped waves that I just did an exposition of. However, when you put in anisotropic, heterogeneous, long wave connections with time delays, Vic and also Peter Robinson showed, you make these eigenmodes unstable. You make the temporal part unstable. They're no longer damped. You destabilize the system. And this is actually what the thalamus is doing. It's creating this unstable 10 Hertz resonant rhythm that's disrupting the neural field. So this is great because if you're doing a task like a working memory task, you need something to come along so that you can swap to a different task. We're constantly multitasking. We're not a stable, damped system. There's something that allows us to jump. Linear systems are fantastic. I mean, as Danny Bassett has shown, they're very controllable, they're very predictable, but they're not very flexible in terms of subtly and possibly randomly going to a different state. So that's what we think these heterogeneous long-range connections are doing. Now, when I was thinking about all of this, I went back and looked at the cytoarchitecture of the cortex. I hope Alan is happy with me here, being a pseudo-anatomist. And the layers of the cortex are very rich. Most people miss, and this is an old drawing, and the old drawings are always the best, that there's 3 layers right through the cortex of unmyelinated axons going horizontally. They don't come out of the cortex. And in the visual cortex, actually, they do get myelinated, and that's why you get striate cortex. But in the rest of the brain, they're unmyelinated. And the cells in those superficial layers are very homogeneous. Well, Singer told me they're like a carpet. You can go from one end of the brain and the other to the other end of the brain, and you'll barely see much of a change. When you get into the deep layers, you start to see the distinct cytoarchitectural changes that characterize the different areas of the cortex. So what's going on here? These big cells in the deep layers come out into the white matter, and that's the connectome that we're seeing when we do diffusion-weighted imaging. We don't see the horizontal connections within the cortex because they're not myelinated. And they're very homogeneous. And then you've got these huge energy-hungry pyramidal cells down in the deep layers. And this beautiful work from Alain Testek shows that these cells are just not going to be triggered to fire in normal resting state. Their resting potential is way away from their firing potential. And exponential EPSPs are tiny. You need thousands of them. When you start to get the cell going closer and closer to its membrane, it bifurcates into this high conductance state. And Max Shine has also been teaching me a little bit about this. And that's why you see this gain curve go up. So when the waves go past, these in the superficial layers, this is my proposition, these deep, large pyramidal cells come into this conductance ready state. And then you have a little bit of convergent input from other connections in the white matter, and then you can fire off this nonlinear instability. So here are these stable damped waves. And now when you get and Gus also talks about ignitions, you turn on these particles connected to the long-range white matter, these beautiful areas of the big brain, and that other people have shown all the beautiful cytoarchitectural differences. That becomes much more apparent, and that mathematically is predicted to then roll the brain over into a different state, very energy intensive. There's a lot of other evidence. I'm just writing this up at the moment. And then it goes back into the low energy damped wave state. So that's this reconciliation. We've got these neural field states where most of the machinery of cognition is being performed. But when you need a state change, you ignite these huge deep white matter projecting pyramidal cells, and you get this sudden chaotic transition to the next state. Yes, so thank you very much for inviting me to speak. I've watched some movies on the plane, and that's why I did the subtitles coming up like this. And I live and work on the lands of the Awabakal and Worimi people in Australia. I pay my respects to their elders past, present and emerging. And those coming to OHBM next year, we're hoping to highlight some of the imaging that Indigenous people of Australia have been doing for 20, 30, 40, 50,000 years. It's very exciting. So thank you very much.
Thank you very much, Dr. Breakspear. We have time for
very much. Yep. Thank you very much.
Chat.
Sorry. Michael, that was a fabulous exposition of and opened, I think, so many new frontiers of how we might consider the spatiotemporal dynamics in brain structure and function. I have a couple of questions, but do we have questions from the floor to start off? All right, I'll start things going. Michael, do you include?
Hello. Can I make a question?
Go ahead.
Hello. First, a wonderful talk, really, really good talk. But I'm from the lab of Pedro, and we are used to working non-linear systems and these kind of things. And I have a question. How can you, for example, if we have this idea that the brain is a non-linear system, and the dynamics should be respond to a non-linear field of some kind. How can you speak of modes in the brain? For example, you cannot, my way to see, the brain wave equation of the brain should be non-linear. So you cannot use, for example, linear superposition to describe the brain dynamics. So how can you still, in this scenario, speak of linear superposition, like in quantum mechanics? I believe that you cannot do this in here.
It's an excellent question. The neural field model is a nonlinear. It's a damped wave equation, but the feedback, the conversion, and this goes right back to Walter Freeman, from the incoming fields through to the cell soma, has a lot of nonlinearities. So that neural field model is a closed set of equations, is a nonlinear model. It's an empirical question as to whether or not you need full nonlinear theory. I came into neuroscience from applied mathematics 25 years ago, armed with all the armory of chaotic dynamics. And I feel, on the whole, the linear approximation performs extremely well. And there's convergent evidence from other people that the macroscopic fields have lost a lot of their nonlinearity. The alpha rhythm seems to wax on or burst on and off with nonlinearities, but a lot of the macroscopic fields are more or less Gaussian and linear. As I showed with that work from the zebrafish, it's just at the level of the macroscopic where the kurtotic nonlinearities seem to disappear. So accordingly, we can motivate our linear approximation of the nonlinear wave equation and do this ansatz into the eigenmodes and their temporal dynamics. And I think it works really, really well. And I think it's only at these momentary switches where you're going to see lots of nonlinearity. I could be wrong, but I've spent 25 years fighting for nonlinearity, and on the whole, I think it's doing the best job. It's having the most fun. It's like party on Saturday night. You go to work, you go to the bank, Monday to Friday as a damped linear system, but on Friday, Saturday nights, out comes the nonlinearities, and that's where all the changes in the adaptation comes from. But I think I'm not convinced we need the full nonlinear theory for everything that we're seeing macroscopically in the brain. It's still an empirical question, of course. Actually, one of the most influential papers is something in biological cybernetics that Pedro published. I'm pretty sure it's you, Pedro. I can never find this paper. There was all this debate about EEG. Was it chaotic? Was it noise or not? And Pedro published this paper, instead of looking at the nonlinear invariance, having a model of the system and saying, could you invert the system? And I think this was with Lopez de Silva, and was the model in a linear, periodic, or chaotic state. Do you remember that paper, Pedro? Or was that maybe that was
Sure,
Fernando?
I'll send you a few of them, okay? But I want to stick something in. I mean, Regal is one of my other students. He's going to present, and he says that he would like me to tell you, because we can later the discussion. You see, he's been testing different components of the EEG, and some he finds that are linear. He's doing tests of linear, but then there are components that are nonlinear, and he has to use the bispectrum to describe them. So he wants me to ask you, if you wouldn't think that maybe you could extend the expansion, to do the spatial equivalent of the bispectrum. See what I'm saying? Higher order, like in the Wheeler-Volterra expansion.
We are doing exactly this. You don't see a lot of bispectrum in the resting state EEG. I've done a project on that. You do see it at times. You see it very strongly in epileptic seizures. So Richard, whose beautiful image I showed, whose beautiful movie, we're doing cortico-hippocampal interactions. And then you see mode mixing, you see bispectra, you see a you know,
Okay.
theta rhythm, gamma rhythm, you see the whole beauty of but it's all
I'll put you.
weak non-spectrum.
No, I'll put you in contact with Rigo. He's analyzed 1,700 intracranial EEG from the MNI, and he's found that if you do like a Fulf type decomposition, the 1 over F, let's say the aperiodic part, is linear and Gaussian basically. But then if you look at specific rhythms with harmonics, then you find a strong nonlinear component. It's interesting to compare what you guys have done with him. Then I'll put you in touch with him. Okay.
Okay.
Also, I have calculated
Tim,
the
that's-
bispectral for the Eigen modes from the James Penn's paper. But I haven't got a good result because there is too less modes to calculate the bispectral. So maybe later, if I can get the high resolution modes, I can get better results.
Okay, so you see they're very interested in what you're doing, but you can discuss, later.
Yeah, Richard and I are going to release a huge package of corticothalamic, corticohippocampal, all simulated with high resolution eigenmodes. Maybe back to Alan had a question. So
I was just wondering, Michael, do you include histological and chemoarchitectural gradients in your neural field models?
this is an excellent question. In the neural mass models we do. In the neural field models, it's already becoming quite complicated when we put the hippocampus in. Sometimes in functional data, we see the emergence of gradients. If you do nonlinear PCA, you will see the gradients coming out of the dynamics. Do we build in the gradients? No, not yet, because it's just too hard for us to do, or me anyway, because you break the symmetry assumptions underlying the Plass-Beltrami operator to get the eigenmodes, and you get second order modes, which then capture that asymmetry from the gradients. So you can do it, but I guess we need data to make us do it. I mean, at the moment, neural field theory has only had a recurrence in the last few years, so there's a lot of work to be done. But no, not with neural mass models. It's very easy to put the gradients in. Neural field models, my brain is already as full as it can get at the moment. But why do you ask? Because they're important, right?
Well, I'm just wondering if you have the exponential relationship that you show, but there's noise in it. Well, it's not so much noise as heterogeneity, and you have different chemoarchitectural and histological characteristics of parts of the brain, which are giving rise to that heterogeneity about the exponential line.
Definitely. I mean, that is a great point. And mathematically, I called it noise. It's mathematically frozen noise, but it's heterogeneity. And there is some mathematics, Carlo Lang and others, looking at neural fields with these heterogeneous synaptic kernels. And you can get some results from it. You can show that the waves are slightly slower when you put the heterogeneity in it. You can start to see that the expression of the modes is no longer uniform, that it gets brighter and sharper in certain parts when you put that in. Yeah, at the moment, most of the focus of what's happening in my group is putting the hippocampus in. And I haven't shown that, but you can do an eigenmode decomposition in the hippocampus and then couple the two structures together.
As a slight tangent, but you should definitely talk with Jordan de Cracker, who's been doing this hippocampal unfolding modeling. I think
our eigenmode decomposition is on Jordan's hippocampus. That's beautiful work. So we're actually working with Jordan. So we've got neural fields of Jordan's hippocampus unfold.
Excellent. The other question that I'm wondering about is, does the neural field modeling inform any discussion of optimal neural mass size? You know, we see blobs and we see cortical columns and about 100,000 neurons, and that suggests a motif that there's something about that scale which the brain in an evolutionary sense keeps on coming back to. And the question that I would ask is, does a neural field modeling approach inform that discussion about what is the optimal scale size for mesoscopic analysis?
This is another great question. Now, what can you see here? Can you see this multi-scale brain modeling?
Yeah.
What do you see there? Yeah. When we go to this beautiful work that Brandon Munn and Max Shine did, you're talking mesoscopically at that second top scale, and that's just when the kurtosis disappears. Now, Mac and Brandon also looked because the fish were swimming, and that was the scale that had the strongest correlation with fictitious swimming behavior. So is that scale down from the macroscopic scale? That's when the neural field model breaks down and you go to that more particular particle network approach, and that's where the nonlinearities come out. So when they're expressed, you're down at that 100 micrometer scale. But it seems that the neural field model still operates on top of that. And that's why the eigenmodes do such a fantastic job at reconstructing task fMRI data. These are group and task average maps, though. They're not the individual maps. But I think this is a very informative analysis, that the mesoscopic scale, where you see a lot of the cytoarchitecture, is just where the kurtosis disappears, when it becomes Gaussian, and which seems to have the strongest correlation in these fish, at least, with behavior. So it does, I think, talk to your question. I don't think it answers your question.
It's a fascinating thing to explore. We have some questions that have come in. Zamzuri Idris, wave energy inside the brain are electric and magnetic waves or EM waves. Opinion or comment on possible presence of plasma waves inside the brain, or even EM waves plus plasma soup solution as magnetohydrodynamics, i.e. brain thermodynamics or plasma brain dynamics. I'm sorry, Zamzuri, I can't.
I think I know. I think I've got a gist of this question. It's like what do the electric fields themselves, if they're very coherent, directly influence neural states? In this theory, the answer is no. That this is all classic neurophysiology with just this mean field approximation. The fields are fields of firing axons that then arrive and go through the classic Walter Freeman approach. The electromagnetic fields that you measure in EEG and MEG aren't directly influencing postsynaptic potentials. So I think that's where this question is getting to. And there is some work out there that's proposing that it does, but I think it's bringing in a lot more complexity, and you need really, really good evidence for this additional complexity, that the fields are not just fields of firing rates, but they're creating electric fields that are also influencing at mean scale. I find that to be a very difficult system to conceptualize and control. But I'm not saying no. I'm just saying let's stick with what I've called the standard theory for the time being.
In fact, that ties into a lot of people that are looking at glial interactions, no? And let's say potassium waves and things like that. I've seen a bit of that, and I agree. It makes things more complicated.
Okay, we have another. Go ahead,
Yeah, but
Pedro.
before that, just one thing. You were asking me, you were saying about history. I think we should mention Paul Nunes, who was the forerunner of much, or if you did, I didn't catch it. Paul was already proposing a lot of the wave stuff very long time ago, especially his book, The Electrical Fields of the Brain.
Yeah, that's a beautiful book. I was really recommend every PhD student in this field to read that. I loved and was inspired by that. And I should have, you're right, particularly when I showed the integral equation, because Paul was working with the integral equation, not with the differential equation. It was Peter and Vic and Herman who took the integral equation to the wave equation, but Paul was working in the integral differential space, but doing wonderful, and his writing was inspired. I met his son, I think. I think it was Paul Nunes' son. Yeah, I should go back and properly acknowledge that. Yeah.
Another question from Eduardo, building on the question about histological data, can any other spatial variation be included in the neural field models, for example, to model neurodegeneration? I guess that's a more general question here, but can you use these neural fields to distinguish between clinical groups?
Well, I mean, you can model neurodegeneration. There's actually some work that Alex Finito's doing, looking at changes in the space in the geometric eigenmodes in neurodegeneration. So you're actually changing the spatial basis functions when you've got changes in the cortical structure. And so that is already how neural field theory can dive into neurodegeneration. To me, what's very interesting in Alzheimer's disease is that the early stages seem to have a predisposition for these large, deep pyramidal cells. So, no, sorry, for the superficial layers. So the pyramidal cells are still there, but they're not getting the superficial inputs from the superficial layers. So they don't get into their high conductance state. It would be like, you remember during the Olympics during the COVID, there was nobody there. It was very hard for the sports people to get motivated in an empty stadium. And I think that's what happens to the pyramidal cells in Alzheimer's disease. They're sitting there, but they're not getting the input from the superficial layers. So they just can't fire.